This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 696, recorded on December 18, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent. Slightly delayed Dixon Despommier, unfortunately, due to the electronics of whatever's going on. Out the window, it's a gray day. It's very cold. There's a little bit of a wind, and it snowed like hell yesterday. We got all of 3 inches here in Fort Lee, but I think other places got a lot, like Binghamton got 46 inches.
We got about 10. I was on a call with someone in Vermont. They got 2 feet.
Right.
Well, we all have 2 feet, right?
That's right. Exactly. I don't understand.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 62 degrees, gray. It's okay with me. It's Friday. The kids will get off school and we'll watch diners, drive-ins, and dives.
What's that?
Oh, I know that show.
Oh.
That's crazy. That is a crazy.
It's a great show. Fridays. This is Guy Fieri. We've talked about this before.
Okay.
Goes around.
He's going to die of a heart attack.
All sorts of just sort of weird restaurants. I actually did a couple in Austin.
Okay, good. From Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's 34 Fahrenheit, 1 Celsius, and pretty gray. We got about 5 or 6 inches here of snow, which is pretty good because it was not enough to mess up the roads too much, but just enough so that I have to have ice on a sledding injury from yesterday.
Oh,
Ooh.
You went sledding?
I took some friends' kids sledding, and when the sled wiped out, I caught us to make sure that that the kids did not, get hurt by sticking out my arm. But of course, that meant that my arm got a bit hurt.
The adult got hurt.
So you have some hill that's suitable for sledding nearby, right?
Yes.
I remember those days. And from Western Massachusetts, Alan Dove.
Good to be here. It is currently 27 Fahrenheit, -3 Celsius, bright, clear skies, and the 15 inches of snow that we got is now cleared off the driveway, but it's still on the other surfaces.
How did you clean it off, Alan? What did you use?
I used the electric snowblower.
Okay.
So the Wubble stands by as a backup these days.
We have a Mount Snowman? Or Mount
Yeah,
Snowplow?
the Mount Snowplow managed to succumb. That's the other thing. The Wubble is really good for taking out Mount Snowplow when it gets too high.
As you know, the Aaron Diamond AIDS Research Center at Columbia University Irving Medical Center is looking for a laboratory assistant manager for their BSL-3 core facility. And we've announced this before a few times. The position is still recruiting. We'll put a link to the show notes. But basically, assistant manager of a BSL-3 facility which works on SARS-CoV-2, among other things. So check it out. And also, ASV is going to have a virtual meeting this year. That means online. I still contend that virtual is incorrect. However, because it's still a meeting, it's not an approximation of a meeting, right?
Right.
So it's an online meeting. I think Alan might agree with me, right, Alan?
I kind of lean in that direction too. In common usage, virtual means online, but I would agree that virtual means almost or something different.
I guess the word meeting means you actually get together, right, physically?
I guess, but
so maybe that's where it works. Yeah. However, I like online.
Yeah, the
anyway.
common usage does not agree with us, unfortunately.
So they're having this meeting online, and the abstract deadline is February 1, and there are you can get an award. There's a deadline for February 3. Just go to asv.org, and you can find the details for that. A couple of brief things, or let's say news, before we start talking about a couple of papers. Now, in the New York Times this week, there was an opinion piece, and there are 3 authors, Jeremy Faust, Harlan Krumholtz, and Rochelle Walensky. And you might recognize the latter name. She is going to be, she's nominated for the director of CDC by Biden, right? And she is at Mass General Hospital, where she's chief of infectious diseases. And this op-ed is called, "People Thought COVID-19 was relatively harmless for younger adults. They were wrong." And so this is an op-ed, but it's actually based on a research paper, which I prefer to refer to. And the research paper is in the Journal of the American Medical Association, JAMA, as we fondly call it. And the authors of the op-ed are also on the paper in addition with others. And the paper is called "All-Cause Excess Mortality and COVID-19 Related Mortality Among U.S. Adults 25 to 44 Years Old, March through July of 2020. All right, so what is excess mortality? It's more than known causes, right? More than you normally expect. What you normally would expect. Yeah, so if you have a bad influenza year, you have excess pneumonia mortality caused by influenza. So they say excess mortality reflects the full burden of the pandemic that may go uncaptured due to uncoded COVID-19, for example, if the death is not coded as caused by COVID-19 or other pandemic-related deaths. And so they said, let's see what we can capture here. And they focused it on adults 25 to 44 years of age. And in this group, of course, unintentional drug overdoses are usually the leading cause of death. And that
to me was one of the most shocking parts of this paper.
Yeah, it is shocking. And the results, basically, it's a statistical procedure. They go through the data, the deaths. But the results are, from March to July, a total of 76,088 all-cause deaths occurred in adults in the U.S. from 25 to 44 years of old, which is about 12,000 more than you would expect, which the expected number would be 64,000. So 12,000 excess deaths. And, among this age group, there were 4,500 COVID-19 deaths recorded. So that's 38% of the excess mortality. And so the idea is that is due to COVID-19. And they can go into that further and say, yeah, this is probably what it was. But, the point is that, well, as we're learning, it's a lot of people.
Yeah.
And as we're learning, anyone who says this group doesn't get infected, that group doesn't get sick, they're just wrong. And they're saying that for other reasons. And we're going to hear about this later from Rich, I think. But the data show that if you're between 25 and 44, you can die of COVID-19. Okay? So.
And that's only the deaths, as we frequently point out.
Yes, exactly.
Yes. Yeah, there are an awful lot of people, who got sick with this and still aren't quite well, and that's not cool either.
Yeah, you get the long-term COVID as well, right?
Yeah.
You can have a mild infection and still get long-term COVID. You get all sorts of outcomes. So,
but 4,500 even if it's not mild or long-term, you can have a relatively short-term illness that puts you in the hospital. You don't die. Yeah. Okay, you're really sick.
And 4,500 people, that's more than we lost on 9/11. Just a little perspective there. That's just the deaths is a huge number of people, and that's not nothing.
Well, that's a confirmed COVID death.
Those are the confirmed COVID deaths. That's why I pointed to that number, is because some people who are denialists would say, oh, well, you don't know that they died of. No, these are the confirmed COVID-19 deaths. And then the other, that's 38% of the excess. And then the argument in the article is basically that what's the rest of the excess? Because you don't expect that many people to die. And a significant proportion of those are certainly going to be people who just didn't get a confirmed diagnosis of COVID-19 before they died.
In fact, the op-ed puts it a little more clearly. Young adults are dying at historic rates. July appears to have been the deadliest month in this age group in American history, modern American history. Over the past 20 years, 11,000 young adults die each July. This year, over 16,000. And so I don't think you can argue to us that that's not COVID related. All right. What else is going on?
No, nor can you say that, it must be harmless in this age group if this is the deadliest era, deadliest month in modern history for this age group, this is really, having an effect.
And this is not political, folks. This is not political. This is the truth.
No,
this is science.
These are the data. This is what we see.
And
so the difficulty is giving that age group a reality check. These numbers mean nothing to those people. There's got to be another way to convince them that they're not supposed to meet in group and wear a mask. And what are you doing?
Well, when you say those people, let's be careful here.
The one member of that age group who is here does find it very meaningful.
But you already did, Brianne,
yes,
right? You already.
I did.
You're highly informed.
I only see one other, group of people. And I am taking lots of precautions. So yes, I did already find this.
Even explain it to your class. Have you gotten any pushback from anybody in your class?
I wasn't teaching as much this fall. I was on sabbatical.
Right.
I had some pushback in the spring from a few students. Yeah. And I have certainly had some emails from students and some questions that have been a little critical of things. So.
Now, the age group that I see mostly not wearing masks and so forth around here is that age group, right? Right. And I think they don't think that they're vulnerable, and that's not correct. And this is not over, folks. We're going up again, as you heard Daniel last night. It's going up again. It's the Thanksgiving surge, and it's going to go on for a while because the vaccine's not going to make an impact for many months.
And just before we lapse into like kids these days type of discussion, I just want to point out that speaking for myself, and I suspect speaking for other folks who are beyond that age group on this podcast, I don't know. When I was in my 20s, you probably couldn't have told me to be careful. It's notorious that there's a perception of vulnerability when you're a young adult and you feel healthy and invulnerability. You think that you're bulletproof. So that's natural.
But you're not.
But you're not. And in this case, that was unfortunately exacerbated by messages that came from normally credible sources saying, "Oh, young people aren't affected." And so I think that's going to be very hard to undo.
So a number of credible people said that, and it's either they didn't look at the data or they had an agenda, some other agenda for saying that. Okay. I think this in either case, it's unacceptable. All right. Look at the data or else keep your mouth shut. And if you have and you want to have an agenda, keep your mouth shut also, because the only thing that matters is people's health in a pandemic. All right. And nothing else. So
keep your mouth covered.
Yes. Keep your mouth.
Indeed. I understand the preachy business, Alan, but you have to get this message to people.
Yes.
And I don't think we can. We don't have a lot of people in that age group listening. And either way, our audience is rather small. I don't know how to do it. And, the New York Times op-ed is not the way to do it. I'm sorry. That's a really limited audience.
It's part of a solution. I mean, that is a loud mouthpiece. It's a much bigger audience than us. And there are certainly and when we say our audience is a particular demographic, I mean, our audience is actually extremely diverse. It may not be huge, but I've been amazed when we get letters from people and wow, they're listening. So I think we do have a lot of age groups, and we have a lot of people who have influence on a lot of age groups. And so this does matter. And I think we've been saying from the beginning of the pandemic that everybody needs to avoid catching this virus.
Yeah. And I think go ahead, Brianne.
For me, one thing that's really important here is that people who are in this age group need to be talking with their friends and other people they know who are in this group and impressing upon them the seriousness. And it's possible that this New York Times piece that does also link to the JAMA piece will be something people can actually share as a tangible piece of evidence of a reason why people should be taking their precautions.
Yeah, and talk to your friends on Zoom, not in person.
And now
Yes.
this Dr. Walensky hopefully will be confirmed as CDC director, and she will be proactive in sending out a message, which the previous director was not. And we're now learning that's because of pressure from the administration, right? So presumably that will not happen in the next administration.
I heard no, it's not going to happen because he's already announced that there'll be a universal nationwide mask up for at least 100 consecutive days.
Yeah, okay.
He said that last night on CNN. And I presume that the people that believe in what he says will do it. And then there's the other group that will just do it out of defiance. They won't do it. That's the ending point. So when does it become an enforceable regulation rather than a strong suggestion? And that's what we're having trouble with, because you can't put people in jail for not wearing masks. Oh, yes, you can. Other countries did that. Somebody got 7 months in jail just recently in some other country for not masking up.
You can in Massachusetts, you can be fined if the cops see you walking around. It's now anywhere you go in public. It doesn't even matter if you're 20, 30 people.
It's not going to work.
If you're out walking on the sidewalk, they could give you a ticket.
But in other states, Alan, that's not going to be
in other
enforced.
states. That's not the case.
Yeah,
and it would be difficult to push down a federal mandate of that.
Yeah, it's not happening. Not in this country. If this went to the Supreme Court, they would say you can't make people wear masks. I'm convinced of it. So,
well, they're not
given the current composition of the court. Yeah.
Yes. So,
any composition, I think that's not a fair thing to say. But I'm a big sports fan, so I've been watching a lot of preseason NBA basketball and some college basketball and some college football. Of course, nobody's in the stands. I mean, a smattering of people compared to what could be there. So at least at some level, they're getting the message. Look, you're not allowed to have a lot of people. And here's the proof. And then you've got cancellation of games. You've got teams that didn't make the playoffs because they didn't play enough games, because they were COVID riddled with people that didn't listen. So why hasn't that spread into the other young people of the same age? That's the fan base.
I don't know.
Well, and let's just in terms of the caseload among young people, let's also bear in mind that is also the core of the working age population. So you're imagining, I mean, a lot of this discussion, people are probably saying, oh, those, 25-year-olds out partying. Well, how about the 30-year-old who's working the cashier counter in order to pay rent, and has to do that and shows up to work and gets sick as a result. So a lot of that exposure in that group, I suspect, is occupational or other, what we would consider legitimate activities, and they just didn't realize that they were at high risk.
Alan, you have your finger on the pulse of the news. Has this particular message? Has this particular message? And we were saying that New York Times is not necessarily the place to get this word out, but has this particular article or message echoed elsewhere that might be?
Yeah, so it's funny. Since the beginning of the pandemic, there have been stories that are often quite powerful stories where, tonight at 11, we'll hear from a healthy 22-year-old athlete who got COVID-19 and ended up on a ventilator, and these stories have been coming out. And I think there is probably a general awareness among a lot of people as a result, because anecdote really sticks with you. Right. I mean, we think in data, but what really gets the message across is, oh, gosh, I saw that guy on the news who, he went from being a track star to being barely able to move. That's powerful. And people have been seeing that repeatedly for months because these cases do come up periodically. So I think that actually has more of an impact than this op-ed. But the op-ed brings it together and says, that's not just a few stories. That's thousands of people in the supposedly unaffected age group who are dead now. This is a disease that affects everybody.
Yeah, don't you think though that the youth of America, they're more social media oriented than they are, the commercial press and news media.
That stuff spreads really well on social media too.
Yes, it does.
Yeah, well, who are the role models that these people would listen to at their age group level that could spread it via that means? And that's what I would say,
I think it's you, Dixon.
It's you, Dixon. Yes.
Yeah.
No, I do not know. I do
no.
not know for the 25 to 44 age group. I have some inkling of it for the high school age group, just because I have a high schooler in my house, and there are these YouTube and Twitch stars who come up a lot. But
hey, I was on Twitch this week.
Cool.
Really?
What game were you playing?
They asked did a live stream about COVID vaccines, and they put us on the front page, and we got 38,000 viewers at one point.
Nice.
Cool. Nice.
It's the third time I've done it. I've was contacted by a couple of people early this year, and they said, we have a lot of people who are gamers, but they should know this stuff. So
Great.
I think that's good to go to different places.
That's excellent.
So I was there for 2.5 hours.
Now you need to get on there and play Among Us.
I'm not a gamer, man. I can. The last time I played a game was about, 15 years ago. I bought this game called Portal, which is a really cool concept.
I keep meaning to play that. I still haven't gotten around to it.
So you have this gun, and you shoot it, and it makes a hole where you can jump into it and come out somewhere else, right? Very clever. And anyway, I'm on my laptop playing it for 10 minutes, and my son comes along. He grabs the mouse. He says, "Get out of here. You don't know what you're doing." And he finished the game in a half hour, That was the last time I tried gaming.
My favorite game was Myst.
Myst was very good in the old days, yeah.
I loved it.
Yeah, Myst was very good. So Dixon and Rich and myself, who are our spokespersons that we look up to? We're just talking about that for this age group. Who do we look up to? Tony Fauci, I guess, right?
Yeah, but as Willie Nelson, if anybody like a popular performer, like George Clooney, for instance, or anybody who makes it big in entertainment, a singer, a musician, an athlete, a retired athlete, not definitely not a playing athlete, but some people that have a cachet with regards to the deniers or the people who sit on the fence. And
maybe. I think you're right, but for me, none of those people matter. I don't care what they say or think.
So say who matters to you.
I told you, I listen to someone like Fauci.
He's already saying this. I mean, Fauci is all over the place, right?
I appreciate that actors and whatever else, they do things that a lot of people see. I do not turn to them for wisdom and inspiration, okay? It's not the way it works for me. I think the amount of attention we place on actors and sports people and celebrities is just perverted in the world, okay? And that's my opinion, and I am entitled to it. So I do not look at them for inspiration, but I would look at someone like Tony Fauci or some other prominent scientist, or in any field, literature, fine, but not a celebrity. I just do not get this. Or a sports player who gets paid so much money, and teachers are struggling and they get paid nothing. It's perverted to me. Okay.
Well, I think Dixon mentioned Willie Nelson, who I think actually would have credibility on keeping one's lungs healthy, right?
I see a sailboat outside the window sometime,
Yeah.
What could help, I think, as much or more than anything else, is a uniform message from politicians.
Sure.
Yes. If you had the federal government, including the House of Representatives and the Senate and the governors and everybody projecting a unified message, if everybody were in the same boat, projected a unified message and said, "We care about you. This is our job." Okay. And in order to keep you healthy, we're going to ask you to understand what the issues are and take these measures. If there were a uniform message, I think that would make a huge difference.
It would.
Yes.
Yeah.
It would.
I agree completely. I think that there are a lot of people who are able to sort of blow things off or not hear things they don't want to hear, and write it off to, well, they all disagree about it, and nobody knows what the right answer is, so I'm going to do what I want. And if there was sort of this nice unified message, that would combat those attitudes quite a bit.
I do think, however, despite my objections, if people listen to celebrities and the celebrities say the right things, that's fine.
Sure.
It just doesn't work for me. Okay.
Sure.
So if some celebrities come out pro-vaccine, I think that's great because people seem to respect them. Okay, fine. But and I agree with the politicians should be making the statements. In this past year, they have not. It's been an utter failure of leadership on all sides. And it's pathetic that people have to die because politicians are having political games. It's unbelievable.
I totally agree. I totally agree with you.
And you know what? If you don't like us being political, we're not. We're complaining that the science in public health was not applied properly. Okay? And if you're saying I'm being political, it's just you think you don't like my politics, and you don't even know what my politics are. You have no idea. People are always writing and saying,
"Come on, Grumpy! I want to hear Grumpy some more here."
Wait till we get to the paper.
Yeah.
You're a liberal. I just want people to be educated and healthy. Okay? That is the job of a government. And this one ain't doing it, folks.
There you go.
I'm a little worked up
Grumpy?
today.
Oh, yeah.
I don't know why.
No, yeah, I hear it is, Vincent, you are disarmingly cantankerous.
Right, yes.
Wow.
So that's a good thing, right?
Disarmingly.
Yeah, it's okay with me.
No, disarmingly means in a good way, right?
Yeah,
Yeah.
You're
Yeah.
charmingly ranty.
that's the official word from Bloomberg. You're disarmingly cantankerous.
But I tell you, this is just not acceptable to have this kind of public health response. I don't get in what kind of world it's acceptable, and almost half of the U.S. thinks it's okay. All right? It's not. This is a serious disease, and they're going to be more. Go figure.
Yeah.
Vincent, it's even more upsetting to realize that it's been around for a long time like this, and we just didn't know it. When
Yeah,
you
I
look back
know.
to 1918 and then the polio vaccine, and it's the same story. We just got more people now, and we can communicate instantly with each other. So
Yeah, that's the thing. Communication has changed it a lot. Yeah, you hear
it
immediately.
really has.
Yeah.
It really has.
But you know what I was thinking? In 1918 pandemic, we only had what? We had one vaccine, right? We had smallpox vaccine, more or less.
Right.
Is that correct?
We have yellow fever also.
I think that was the that was the 40s.
No, that yellow fever
And
was
rabies?
based on.
We had rabies.
No, because we didn't know what
Pasteur.
in 1918 they didn't know what viruses
Pasteur.
were.
I'm thinking about Pasteur now.
Actually,
Come on.
Yeah,
They
Pasteur.
just barely knew what viruses were.
As I barely knew, and they barely had vaccines, and there's no way they could have whipped off a vaccine in a year, right?
No.
Not happening.
No,
Such
no.
a stark contrast. Okay.
And really what resolved the 1918 pandemic was basically that everybody who was going to die of it did, right? I mean,
Just about.
and you can see in the life expectancy statistics of human history, we
right.
get longer and longer and longer lives, and there's boom, and then we get longer and longer. Wait, what was that boom? That was 1918, and that was the flu. That had a massive impact.
That data, or those data, are always so dramatic to show students. That sort of makes all the points I need to make in that class just by showing that.
And
Second.
the first thing people think when they see that graph, if they're not microbiologists, is they look at this. Oh, that was World War I. No, that wasn't World War I that did that.
It's the first slide
Not
I show in
even
my vaccine lecture. Yeah.
Yeah.
By the way, Alan,
do you show the slide? Do you show the picture of the what was it, mortality records or something in some state, where they got a bunch of books up on a shelf that are the yearly mortality records
that's a good
for a given state, and 1918 there's like 2 volumes, and each of them are 3 times the width of all the other flanking volumes.
Cool.
I'll see if I can find that.
Yeah, find
I
it.
have to look for that.
Alan, the FAA approved the Pfizer vaccine?
Yes, and I'm not misspeaking. I do mean the FAA approved the Pfizer vaccine. So a week ago, the FDA approved it, and it is now. You may not be aware of this if you're not in aviation, but pilots and air traffic controllers have to get medical checkups, and they can only take approved medications and vaccines. And if you're taking something that's not approved, then you need some kind of special procedure to get your medical issued. So the FAA, when this was approved, had to look at the data, and they have an aeromedical board that does this. And they said, oh, yep, this looks legit. It looks like it doesn't affect your ability to fly or control air traffic, but you have to take a 48-hour break after each dose of the vaccine. So you can't fly an airplane or control air traffic within 48 hours of your of your shot, because they just want to be sure that you're not going to have some delayed, and the widespread understanding is that especially after the second dose, people feel pretty crummy and probably shouldn't be doing taxing activities. So I thought that was kind of interesting. That
it is. It's cool.
There's another level of approval that was necessary here.
I didn't realize that. So probably Moderna's vaccine will get the EUA from the FDA today, right?
That's what I hear. Yeah.
Brienne, did you watch the whole thing yesterday?
I did not watch the whole thing yesterday. I had meetings and then I had to go sledding. So
there you go.
Yes.
Then you went to the emergency ward.
Very good. All right. So we have a paper to do, which I originally, here's this history. So on Sunday, Amy sent this to me and she said, don't you dare do this on TWiV. It's trash. And I said, okay, I think that's a good idea. And then this thing hit the news, it hit the Twitters, and people are going nuts. And I said, we have to do it. If anything, just tell people why it's not a good paper and why the conclusions are not justified. So
I think we should start by pointing out it's not a paper.
Yeah, it isn't even a paper.
Preprint. It's on bioRxiv, and we only occasionally do these. They have not been peer reviewed. Anybody can put anything on bioRxiv.
I would be surprised if it's published in any form unless the experiments are done that are needed, which we'll get to.
This is a complete redo. Yeah.
But it's a bioRxiv, and I would normally not do it because I don't want to give it attention, but it's gotten so much attention, and you have to understand why it's unjustified. Starting with the title, completely unjustified. SARS-CoV-2 RNA reverse transcribed and integrated into the human genome. So what do you think when you hear that? Oh my gosh, patients must have the genome integrated in it. Well, it's not. There's no data on that, as you will see. Anyway, it's from Zhang, Richard, Khalil, Wolgram, Ma, Young, and Yanish, Whitehead Institute, Harvard, MIT. Some two PIs, Young and Yanish, well known in their fields. They are not virologists. And this is problematic. And, the thing is that some people think this is brilliant, and reputable people think it's brilliant. They've been quoted as saying as such, and others say it's not in the reason. It's not as technical reasons that are due to interpretation, which we'll go over. And it's kind of disappointing that people didn't read it carefully and say, you can't make this conclusion.
And some of the reputable people who are being quoted, I think, don't really have the specific technical background to see the flaws in this.
Exactly.
Yes.
Now, go ahead.
I just wanted to say that the other thing that scares me with the title is that I have been answering a fair number of questions from a lot of different angles with people who are asking me if there's any way that the mRNA from an mRNA vaccine can be integrated. Right. And so you could also read this title and it could make you think that something is a problem with the mRNA vaccine. Spoiler alert, no, it will not.
There's no evidence that would happen here. From this paper, there's no evidence, as you will see. So the driving force, apparently, according to the authors, they wanted to know why there's persistent RNA shedding, which, in my view, and Diane Griffin has said this years ago, after an RNA virus infection, even an acute RNA virus infection, RNA resides for a long time. We saw that with Ebola. We see it with many RNA viruses. There's no infectious virus, as we've seen in a number of analyses, yet pieces of RNA continue to be present. They're pieces of RNA. So they're thinking, wow, it must be in the genome, and that's what's making it. And I just think that you're trying to find a solution for something that doesn't need one. Okay, so what did they do? They said, okay, maybe the genome is being reverse transcribed. Cells contain reverse transcriptase. It is not only a retroviral enzyme. In fact, reverse transcriptase, which copies RNA into DNA, was around long before retroviruses appeared on the planet. It's a cellular invention. And retroviruses happen to grab it and become retroviruses, but we will have it in our cells. Bacteria have it. Archaea have it. Okay, it's not unique to viruses. So they're saying, well, this cellular reverse transcriptase, maybe it's copying the SARS-CoV-2 genome and bits are getting integrated, and then those are getting transcribed, and that's why we see this long-term positivity. This is so perverse in a way, right? From my view, the virus is killing cells. It's a lytic virus. And so who cares? Even if it did integrate, the cell would die. And anyway, they haven't even looked in people. So what they do is they look at sequence reads from high throughput sequencing that's been published, and you can go and you can download these sequence reads. And here's the tech. When you do high throughput sequencing, say of SARS-CoV-2, you take a sample, you extract the RNA. Typically, you fragment it, and then you reverse transcribe it to make DNA, which you then amplify by PCR, and then you sequence it. Okay, that's the rough. And then you get a lot of reads, and you throw out the ones that don't look very good, and you keep the rest, and you assemble a genome or whatever it is that you're looking for. And you put all these reads on servers that other people can look at. So,
and note that you don't put the reads in right at the very end of that process. Some of the reads that you may have thought didn't look so good still get put up.
Yeah. So you can put a lot of garbage up. So
what's in the database is raw data from shredding all the books in the library and reading a sentence or a few words from each, and in no particular order. Yeah. And that's all in the database.
Now, remember, I said, how do they do this? Is they take RNA and they reverse transcribe it. The RNA is extracted from cells. It's got cellular RNA mixed in with it. It's not pure viral RNA. It's a nasal swab, right? They extract. It's going to have cellular RNA. And guess what happens during reverse transcription? The enzyme could copy a little bit of one RNA and copy a little bit of another and make chimeric molecules. Happens all the time. So what they did here, they went into these high throughput databases, and they found chimeric transcripts, human and SARS-CoV-2, joined together.
So far, if you're familiar with this technology, nothing surprising has happened.
Nothing.
Right.
And in fact, if you look over on Twitter, there are two people on Twitter that have done a good job, Ari and Halvors. And I think it was Halvors who said the chimeric reads are almost certainly artifacts, and this needs to be ruled out for the natural integrations. And artifacts generated during the preparation of the sequencing library.
So when you do lab work and you have weekly meetings, doesn't this come up regularly as a caveat?
Of course. Should.
It should.
So it should have.
In a pandemic, Dixon, everything's out the door. Everything's out the door. Pandemic science is orders of magnitude less rigorous than normal science, in my opinion. And what I've seen in this past year.
There's a lot of weak science. There's some incredibly good science that's been done.
Yeah.
And that we've talked about. But there's a lot of weak science, and it has intersected with the rise of preprint servers, which initially people thought, oh, great, open science, we'll share our data, and look how well it's worked for the physicists. The reason all these are called RXIV, in the name, is because archive was the original preprint server for physicists, and they've been doing this for years and years in physics. Yeah. And it works great for physicists because they can share their data, and then somebody comments on it, and your math here should be like this, and it makes for better science. But the difference is that in physics, none of these preprints are going to get picked up and hyped by media outlets and cause people to go browbeat their doctor to prescribe them more quarks, right? It's not. I'm not saying physics is unimportant, but what's being covered in it is not going to change anyone's day-to-day behavior. In biomedical research, and especially in epidemiology, there's this issue that people could get, since these are public servers, people can get this paper, looks like a paper, and say, oh, well, this shows this. Well, if you understand what's going on in this paper, but most of the people who have access to it don't, then you see that doesn't really prove that. And part of the peer review process for a traditional journal is that you have to provide enough data to support your conclusions. that barrier is not there in preprint servers. And that's what we're seeing. And now that we've got a pandemic going on, people are dumping stuff like this into the preprint servers, and then it gets picked up by news outlets. And now we on TWiV have to go through and explain, no, this is.
So they have these chimeric sequences, okay?
Yeah.
And the problem is they have a piece of human sequence next to SARS-CoV-2. And that's not good enough because it's an artifact. Let me read a few more comments from Twitter. Show me genomic DNA sequence spanning the integration junction. Okay, so if you're saying it's integrated, show me the integrated DNA. Either do a southern blot. Hey, folks, you remember those? They still work. People still do them, showing integrated SARS-CoV-2 with human sequence on both sides. That's what you would need to prove, and they haven't done that here. They haven't even looked at cells from patients. They just looked at this database, which is full of artifacts. And this other comment.
Correct me if I'm wrong, but nobody picked up a pipette in this paper, right?
Yeah, they did do some experiments, which we'll get to, but they are just.
Oh, that's right. Yes, right.
Just irrelevant, basically. We'll get into that. So. Again, continuing from Halvor's, you found RNA-seq reads that look like chimeras between cellular genes and viral genes. Congrats! I can show you mouse-human chimeric reads. I can show you bacteria-human chimeric reads. I can show you things you wouldn't believe. If you dig through the trash heap of unaligned reads in any high-throughput sequencing data set, you will see things that make you believe in demons. Everybody involved in any sort of high-throughput sequencing project should be required to spend some time listening to an embittered bioinformatics guy rant about sequencing artifacts. That's just great. Yeah, I just love it.
I love all of this, and that does make me really happy to think about, the mouse-human bacteria hybrid.
If you went through these databases, you could find hybrids with almost any virus that was originally in the original sample. You could find influenza and others. And by the way, we on TWiV have talked about endogenous sequences of RNA viruses integrated in mammalian genomes, bits of RNA virus genomes in the germline. Okay, that can happen. They don't reference a single one of those papers here. They are completely unaware of that literature, and that's inexcusable. If you're going to publish something like this, you need to go in the literature and go, oh, it's been shown to happen in the germline. But of course, in the germline, they got both ends and they show that it's a bona fide integration. And I can't believe they didn't reference that work at all. I mean, what are you thinking, folks?
I thought the reason why they put it up here is to elicit reviews so that they can correct whatever they think is correctable.
The problem is that by the time you look through what they've got here, there's nothing to make a paper out of. So the idea of a preprint server is that you do solicit reviews for something that you're going to move toward being a paper. But there's this artifact issue, and I realize the reason I forgot about the wet lab experiments is because they're so they're kind of bonkers, right?
They are bonkers. So let me tell you the wet lab experiments. So
can I say one thing?
Yes, of course.
Sorry. And this is more of for our listeners who are not specialists at all. Just to be clear, what can happen here is that the enzyme that is reverse transcribing, taking RNA, making RNA into, or making a DNA copy of the RNA, can fall off the original RNA template. That happens at some rate, and then it will re-anneal. It will go back on, but it might pick the wrong RNA template to bind back onto. And that's how you get
right? There's just RNA. There's no wrong or right.
Right, exactly. But it will bind to a different one, and that will result in this chimeric product. And so this is all just about the enzyme coming on and off of an RNA template.
So if you did this analysis and you thought, there's some evidence, even though it's flawed, do the right experiment to prove it. And the experiment they do is completely bonkers, as Alan said. They take
and if you don't know what the right experiment is, you should go to the literature and find colleagues who do and talk to them. Hey, we found this thing and it looks kind of weird. What do you think? And that's where your bioinformatician would say, oh, yeah, that can happen. Here's how. Here's what you'd need to do to show if it's real. And then you go and do that experiment.
There must be tons of people at the Whitehead that could answer that question.
Oh my gosh, the
without
place is
going to
lousy with
literature. You can just
bioinformaticians.
go
They've
down
got
the
it coming out
hall
of the
and
wall.
ask somebody. Ask the janitor.
It's hard to figure out. So what they did, they took cells in culture, HEC293 cells. I could care about HEC293 cells in SARS-CoV-2.
You could give a heck.
And they overproduced a cellular reverse transcriptase. It's encoded in an element called line 1. We all have these RTs in our genome. So they take the gene which has been cloned, they overproduce it in this cell, and then they infect, and lo and behold, the SARS-CoV-2 RNA is reverse transcribed to DNA. I mean, how does that prove in any way that what they're thinking is actually occurring? Yeah, if you put RT in a cell and you throw RNA in, the RT is going to copy the RNA into DNA. It actually gets integrated as well, but none of this is surprising. It's totally artificial and bears nothing on their idea that this is happening in a bona fide infection. I mean, it's embarrassing.
So the RT is reverse transcriptase.
Yeah,
Yes.
and the RT is in the cytoplasm of the cell, which is where the viral RNA is. So why are we surprised that this is happening at all? I fail to understand why they did this experiment. Instead of taking someone with COVID-19, taking a little nasal swab, extracting the human DNA, not RNA, and doing a Southern blot and seeing if there are SARS-CoV-2 sequences integrated into the genome. It's so friggin' easy to do that experiment. Why didn't you do it, folks, instead of overproducing enzyme in a cell and showing that it gets in? That is just a crazy experiment. And then even crazier, they say, hey, let's see if cytokines can induce reverse transcription in cell. Maybe that's what's going on when the virus infects a cell. You got cytokines all over and yeah, so they add cytokines to cells, and I don't even know why they picked the cells that they did. You know, they picked myeloid cells, microglia, T cells, and they add cytokines, and they show that the line 1 RT goes up. Again, as far away from the original idea as you can get. It does not support it whatsoever.
I have a theory about why they picked these particular cells in experiments, which is they had them sitting around.
Oh, that's right.
Oh, that's a good reason.
That's right.
That's a good reason.
That's what Amy would say. They
That's
opened
the
the
simplest
freezer.
explanation. Pull something out of the freezer that can do this.
Pull something. Amy said last time or whenever, she said, you just open the freezer and you decided to use what you had in there. Maybe next time you should think more and close the freezer before you do it.
It's a science frittata.
What's that, Dixon?
A science frittata. That's how we make a frittata at our house.
Yeah.
We go in and pick up everything and throw some eggs in it, and then you got a frittata.
So if I was going to be exceedingly generous, and say that I bought any of what I'm seeing here, the question that I would have for them is, you overexpress reverse transcriptase and you show that SARS-CoV-2 RNAs get into the genome. What about all the other RNAs in the cell? Why is this special to SARS-CoV-2?
Sure.
And why is it not happening with every other RNA in the cell? And why is, life possible?
Well, in fact, if in their experiment, Brianne, if they had looked, they would have found other RNAs being reverse transcribed and integrated. Absolutely. Because in fact, in our cells, routinely, mRNAs are reverse transcribed by line element RT, and sometimes they integrate somewhere else and they cause issues. We know that happens. It's exceedingly rare.
Right.
But if you massively overexpress that reverse transcriptase, then you can get all kinds of RNA integrated, and that's exactly what they see, but they only looked at the one they were interested in.
Right.
Now, again, the experiment they should have done immediately if they really thought these chimeric transcripts meant anything, and they don't. They're an artifact. Take some DNA from an infected patient and do a Southern blot to look for both ends of the integrated SARS-CoV-2 and flanked by human DNA. And they did not do that. And I can't see this being published without it. I don't know why you would.
No.
And it's too bad we have to spend time on this because there's so much other good stuff out there that it's actually good science. But so many people.
But it's important. I think this does teach one interesting thing, which is you shouldn't go trolling around bioRxiv and looking for results that are going to have meaning. I mean, we will occasionally highlight something there that looks promising, but when something gets picked up and hyped the way this has, you've got to take a a very skeptical view of that, and it's an artifact until proven otherwise. And in this case, I don't think they're ever going to prove otherwise.
And by the way, I actually don't care if this integrated into my somatic cells.
There's
all
that
somatic
too.
cells die within a few days, right? And so people are saying, oh, like Brienne said, the mRNA is going to modify my genome. You know what? If it modifies the genome of a skin cell, I don't really care. Or a respiratory epithelium, because they're all dying. What I would care about is...
especially ones that have been infected by SARS-CoV-2. They're dying real soon.
They're going to die. And even if it somehow, the RNA left and got into another cell that's not infected and integrated, I don't care. It's going to die in 3 days. Now, if it went into my germline, I might worry, but I think that's not happening. We don't have evidence for this. So, folks, and Alan, the issue here is that the press doesn't know how to handle this, right?
Yeah.
So they call someone up and they say, what do you think? And they haven't read the paper and they go, wow, look at the title. Wow, that's pretty cool.
Right. And the scientific community doesn't handle this very well because, in biomedical research, I think a lot of folks still haven't gotten used to the concept of bioarchive. And so they look at it and they say, oh, wow, this is a paper. It's published. And there is a bias to assume that there's some level of validity to that. Well, they wouldn't have published it if it didn't have something.
Yeah.
Well, no, they haven't published it. It's not published. It's a preprint. And so there's that going on. And yes, the process for a reporter on seeing something about this would be, oh, okay, I'm going to call up some sources who weren't involved in this work and ask them about it. And of course, they haven't heard of it because there are gazillion papers a day on a preprint server. And they take a quick look at it and they say, oh, wow, this is really interesting. So-and-so of such and such said this is really interesting. And so that, that then becomes the story, and it really shouldn't have been.
So isn't it obligatory to submit your paper for review at the same time that you put it up on the server? No. So this paper may never get in front of a real review session then.
It may not.
It would be best if it didn't. Unfortunately,
I would expect if this paper,
as we speak.
No, if this paper in its current format was submitted to any reputable journal, the research editor would look at it and reject it out of hand. It wouldn't get to peer review. It would just be, okay, guys, just go work on this.
I would hope so. However, in the COVID pandemic, I think these journals are dying to get anything COVID. And if you review this and you don't reject it outright, you're doing a disservice to the field. This is severely flawed. What do you think, Rich? You got anything to say here?
You guys have said it all, and I agree with what you said.
Okay. Is there anything else? Okay, so, unfortunately, this even got covered in Science. I think there's a lot of pressure that if somebody's covering a lot of this story, you got to cover it too.
Well, and guess what? Now it's been covered in TWiV.
Yeah, but we trashed it.
I mean, yeah. But yeah, the story is being covered, so you have to talk about the story that's being covered.
This reminds me a little bit of that bit with in a hearing, one of the senators holding up an observational study that was flawed about hydroxychloroquine and holding it up to Fauci and saying, but it was peer reviewed. And Fauci says, I don't care.
Yeah.
And this is a study from reputable researchers from a reputable institution. Okay. Trust science, not the scientists.
Yeah, these are people who I believe have done. These are people who I believe have done good work, but this is not it.
So yeah, even reputable people can make missteps, because people always say, well, this was said by a reputable person. And so I don't care if they're reputable. That's a good start. But if you examine it closely, it's not reputable. And often the public does not know how to distinguish between opinion and data. If, for example, someone this week said, well, this idea that more virus makes you sicker, right? The whole dose severity thing we've been dealing with. It was in this paper in the New England Journal, and he sent it to me. He said, here are the data. And it's an opinion piece. There are no data. And I don't know, people probably don't know the difference. Sometimes you can express your opinion.
We're used to it. We've been doing this for a long time.
That's why we're on the air, to show you what the difference is.
Okay, enough of this.
That's our job.
Enough of this.
Yes. Also, please don't scroll down and read the comments.
Oh, no, don't read comments on anything like this.
Yes.
No, never. You should just listen to TWiV. By the way,
and we might point out that this has been picked up by people who have agendas to be anti-vaccine or COVID-19 is a hoax. The tests are all false positive. I mean, all of this has been making hay out of this preprint, and that's.
As a matter of fact, the authors ought to take this down.
Yes, they really should
take it down. Yes, authors, please take this manuscript down.
Please.
It's an affront.
Sounds like Ronald Reagan talking.
Yes, tear down this manuscript.
No, tear down this manuscript.
Yes. Or to Gorbachev, I think.
Good,
tear down
maybe
this
a
wall.
good title. Tear down this.
Yeah, there we go. Tear down this manuscript.
And another issue that I've encountered this week is encompassed in this email from David, who writes, "It's been announced this week that variously a new strain, variant, or clade has been identified called VUI 202112/01, which has been described in the, quote, popular press as deadly or more virulent. Whilst I take stories from such media as apocryphal and designed to generate heat rather than light, I wondered if you could elucidate on the differences between these terms and their significance. Strikes me that miscommunication is almost as damaging as any other aspect of this pandemic. It needs to be challenged, but with national leaders like the POTUS and indeed our own PM in the UK, muddy waters run deep. The latest mutation is reporting as affecting the spike protein, and given that this is the target of various vaccines, what are the likely consequences with regard to vaccine efficacy? I'm only a layman. I guess you could say I'm just a layman, right? But thanks to TWiV, and I hope a modicum of reason, I feel better able to counter some of the muddled thinking that I encounter on social media, not the foil hat conspiracy theorists who I fear are a lost cause, but the sort of thing generated by misunderstanding and misinterpretation of mainstream media." That's actually put quite well.
Yep.
The muddled thinking.
I like the foil hat metaphor. It's a
It
good
is
image.
good. Yeah. All right, so you want to learn about this variant, go to GISAID, GISAID, which is a sequence database that every time a SARS-CoV-2 genome is reported, it goes here. And here's the entry. UK reports this new variant, which I just read before, is defined by multiple spike mutations. It's a variant. It is not a strain. A strain is a virus with a new biological property. There have been no new properties ascribed to this isolate other than sequence differences, which is not enough to make a strain. So it's a new variant.
Nor have we seen any other new strains of SARS-CoV-2 that anybody's demonstrated to be biologically different.
None.
That hasn't happened yet. I'm not saying it couldn't, but
no, exactly. Yes. So this variant is defined by multiple spike changes. There's a deletion of 2 amino acids in a couple of different places. Then there's individual amino acid changes. And they say there are 24,746 viruses from the UK in this sequence database with a collection date since the 1st of November. A small fraction of them, about 6%, share several of these mutations. Based on evaluation of effect on virus structure and function, the most relevant might be an amino acid change at 501 on the spike protein and the deletions. They could contribute to changes in binding or antibody binding. The other changes, not clear what they would do. However, the detailed effects of these mutations remain to be determined fully. Nobody's done any experiments to determine the effect of these changes on any property of the virus. Yet, when a journalist calls up a scientist and say, "What do you think?" And the scientist says, "These could have an effect on virulence or transmission or antibody neutralization," somehow that gets translated into it's making the virus more virulent. And that's what circulated. And a dozen people this week said to me, "What do you think about these mutations in the UK that make the virus more virulent, more transmissible, more resistant to neutralization by antibodies?" None of that has been shown, folks. It's speculation. There's no paper on any of this.
One of the things that you said there, Vincent, is when a journalist calls up a scientist, and I'm strongly suspicious that in this case it was the other way around, that a scientist called up a journalist. Because, this kind of stuff is going on all the time.
Yeah.
Okay. And this particular one hits the news. I got to wonder why.
Well, this one, okay. So this was mentioned. Actually, it became part of, let's see. Somebody told the House of Commons.
Yeah, Matt Hancock.
About
yes.
this. Matt Hancock told the House of Commons. So this was in somebody's testimony before the House of Commons. They were asked about this, and it came out in the course of that. So it became a matter of public record.
So there's an article in BMJ which I linked there, and it goes through this where Matt Hancock told the House of Commons on 14th December that initial analysis showed the variant may be associated with the recent rise in cases in the UK, and that gets translated into is associated with, apparently.
And the problem here is a misunderstanding between most people and scientists on the issue of probability. Could this happen? Well, sure, it could happen. That's not a fair question to ask a scientist. Anything could happen. We've seen all kinds of stuff happen.
Yeah.
So what we say, could this make it more transmissible? Sure. Could it give it the ability to fly? Sure. It's anything's possible. And we really, as scientists, we really inhabit that world. But for journalists, they're thinking the way you would talk to people in other fields. If you talk to an engineer about could this happen to somebody's car, and they say, well, no, that's not possible. Well, it's just because to an engineer that can't happen.
Yeah.
That's actually within the range of reality for them. So the other thing that gets people's attention is it's infected like 11. It's been detected in 1,100 people, in a whole bunch of cases, and so that causes this immediate assumption. Wow, look how widely this strain has spread. Well, okay, it's not a strain; it's a variant, and the reason it spread to those people is most likely that they caught it from other people who had it. And so you had a particular mutation that was introduced, and then it's like, these celebrities who are famous for being famous, right? It's a question of who infected who and which particular virus got spread furthest, and that is a matter of luck.
Yeah.
It's not about the virus being better at spreading. It's that somebody who got infected with this mutation went to choir practice or went to a bar or went to, some other thing and spread it to 100 other people. The one we've been talking about for months, the D614G, right?
Yeah.
That one, I think, was in a couple of super spreader events early on. And so it's become the predominant strain or predominant variant, right?
Super spreading is the rule.
It's not the predominant variant because it's so much better at infecting necessarily. It's the predominant variant because it's the predominant variant. So now if you catch SARS-CoV-2, you're almost certainly going to catch D614G because that's what's circulating, right? And this is the same way. And you see that this has spread in a particular area of England, and there have been a couple of cases scattered around. But I would bet a beer that if you had the ability to do detailed case tracing on those cases, you could probably find the links that connect this variant back to the original source. And it's just a question of those chains of transmission really go that far.
By the way, Alan, you don't get infected with a mutation, as you know.
No, you don't.
But you did say that, so I just feel the need to correct it.
Right. You get infected with a virus that may carry a particular.
But I think that's a perfect point. The super spreading is the way most of the transmission occurs. And if there happens to be someone carrying this virus with this sequence, they're going to give it to a lot of people, right?
Yeah.
Okay, one last thing before we move into emails. This is not virology, but it is science. It's computer science. And I think as the pandemic fades, we're going to be haunted by this story for a long time. Apparently every important computer system in the U.S. has been hacked by someone and has been breached. And it was first, revealed by the Washington Post on the 14th of December. And the more people look into it, the more serious it seems to be becoming. And I know we have listeners out there that are far better than we are at understanding this. So if you'd like to send us some brief summaries, but this scares the hell out of me. I mean, the power grid infrastructure has been infiltrated by these individuals. So all the power in the U.S. could go out, I guess, right? People don't seem to be too upset about this, but I think it's pretty serious.
This is a pretty big deal. Yeah.
So last night on the public broadcast station, they had several experts on various aspects of security, cybersecurity, and one of them said that you can rest assured that none of the top secret, you're not allowed in unless you have a special password, none of that is on the internet. So there's no way they could have hacked into that inner sanctum. But there's no evidence that they actually altered any of the data that they downloaded and then went back, like change a decimal point on somebody's bank account and make $1 billion out of $1 million, that sort of thing. And I haven't done anything to it yet. They're just information gathering, which I agree with you, Vincent. I don't like people looking into my private life.
But they've put back doors into these otherwise secure computer systems, which means they can go in anytime and muck with it. So that's not good, right?
So can't you alter that quickly with a cyber?
No. They said on the same news report last night, Dixon, they said, you can ultimately clean this up, but it's going to take a while. I mean,
exactly.
We're not talking months either. The installed base on some of these systems, when we're talking about critical infrastructure systems, the turnover on those is glacial, because they have to go through so many approvals and such a process of integrating the whole system that you end up with something that's, some computer system from 20 years ago is still running the critical system, because updating it would mean rebuilding the entire critical system. My mom actually worked on a project. She worked for a company called Air Inc., which you've never heard of if you're outside of aviation, but they're very integrated into air traffic control and airlines and aviation. And they had this huge contract that went on for years and years that was part of renovating the entire U.S. air traffic control system. And they were replacing stuff that was, reel-to-reel tape-driven computer systems that were running the air traffic control system into the 90s, and that's the kind of thing that then, if that's been hacked, now I don't know that ATC has been hacked necessarily, and there are safeguards in a system like that, but the electric grid is a good example. I'm certain power plants are dealing with the same thing, where there's some 40-year-old technology that's in there that's vulnerable because of the way things have been added on. And it's going to be a big project to try and fix something like that.
Yeah. I just wanted to bring it up because it scares the hell out of me. So, I guess similar to how viruses scare people who don't think about them very often, this scares me. All right, let's do some email. Brianne, can you take that first one?
Sure. Kathy writes, while the panel was discussing the dosing errors involved in the Sanofi trials, I began to wonder about dosing in general. My questions. How is it possible to dose a 100-pound female and a 300-pound male with the same efficacy if all the vials of vaccine are a standard dosage? And have any trials looked at the antibody response and duration in light of the standard dosage? Many thanks. So I believe the earlier phases of the trial do some dose escalations. And so that sort of gets at your second question about what types of responses have been examined. The duration, of course, with the SARS-CoV-2 vaccines can only be examined over the amount of time we've had those vaccines. Though I would imagine that certainly can be done and has been done in animal studies and with other vaccines. I think I have an answer for the first question, but I will let somebody else who feels more certain of their answer take a swing first.
Let me just add before you do, let me just add to the second part. So they did do some dosing, but also they selected a dose and they used that for the efficacy trial. And then they measure antibody and T cell responses. And as you know, they seem to be on a par on average with what you see in a natural infection, right? Same kind of levels of antibodies in T cells. So that's why we think we're at the right place. And in animals, yes, they can determine more easily what levels, say, antibodies are needed for protection. And the human results seem to be in line with those for what that's worth.
And all of those dosing studies are done with a mixed population of individuals, balanced males and females, people of all different sizes and shapes. Actually, Brianne, I was going to ask you about the answer to number one, but I have an idea. I'm making this up.
Okay.
And that is because it's a good question, you know? And my idea is that, you know, in a normal drug study, you're thinking about something that is going to become systemic. Okay. So you inject something or give it IV or take it orally, and it's distributed throughout the system. So the concentration, it seems to me, is going to be influenced by body mass. In this case, you're injecting something locally. And it's then, as I now understand it, likely picked up locally by antigen presenting cells taken to local lymph nodes. And it seems to me that that's going to be less sensitive to something like your total body mass. That's the way I think of this, but I'm totally making that up.
I think that makes a lot of sense.
I think if you're larger and you have a certain concentration of, say, B cells per milliliter of blood. If you're larger, you have more blood, right? And your system will make the extra B cells that you need, right? Because it's just a matter of cells proliferating. So you go in with the same inoculum of vaccine, and then your body makes up for it depending on your size. I think that's basically what you said, Rich. You need more drug because our body is not making an antiviral. It's just using what you put in. But our immune system can manufacture antibodies and T cells at the levels that it needs. So that's why it works with both. It's a good thing because otherwise we'd have to vaccinate by mass, as we do with some drug treatments, like ivermectin is mgs per kilogram, right?
Yep. No, that was exactly kind of the line of thinking that I had, was that, you know, there's this amplification part of the immune response to any of these that is not there with many of the different sort of compounds that we give as drugs.
And so that's it. Yeah.
You can have, and so the amplification will sort of amplify the dose or the immune response to scale your size. But
great questions, right? Really good questions.
Yeah, excellent questions. And it is true that for drugs, this is a major issue because the drugs are not being, you're not trying to get your body educated about a particular threat and then let the immune system handle everything. I mean, if you need a particular dose of a statin compound to control your blood pressure, you need that to be maintained at a particular level in your bloodstream constantly. And it's going to make a big difference if you're a 100-pound female versus a 300-pound man. But the immune system, what you really need to do is just give it the memo. You know, you say, hey, prime it. This is a threat, just so you know. And then the immune system spreads the word throughout the body.
Alan, can you take the next one?
Sure. So we're up to Dominic.
Yeah.
Dominic writes, my wife and I have watched every episode of TWiV since March because it's the most informative, science-based source of information we've ever seen. Of course, we forward along your shows to spread the word too. All of our age group are now 70 and above, with many people on meds for the various age-related conditions of high blood pressure, diabetes, high cholesterol, etc. Many of these folks continue to travel by car or plane and rationalize their safety risks. Have you ever seen any science-based studies which have documented the airflow direction, filtration methods, effectiveness, and level of risk of others on the plane? If others on the plane are infected with COVID-19. If your seatmates are shedding millions of particles, particularly without masks, say during mealtime, it seems as though those overhead air nozzles could simply be blowing the virus everywhere before the air is recirculated in the filter system. Friends are taking an 11-hour flight from Massachusetts to Hawaii to escape the high COVID numbers. Sounds great if it's safe. All right. So yeah, I have not seen peer-reviewed data specifically looking at planes. I have seen repeated mention of the fact that we don't have any reports yet reliably saying that there's been a super spreader event on a plane. Please, anybody jump in if I'm wrong there. Going into this pandemic, one of the first things that I thought about flying on the airlines was, gosh, you're going to cram a bunch of people into an airplane that's not going to go well. In practice these days, they're often not cramming a bunch of people into the airplane. The airlines would like to have more passengers, but there's not the demand, and so they end up with these half-empty planes that are wrecking them financially. But so far, there has not been a lot of evidence of spread on planes. And we got a letter a while back from an airline pilot who gave a very detailed description of the purification system built in and how that works. And it seems like it's a lot less risky than you might think, but it's certainly not zero risk. I certainly understand why the people are flying from Massachusetts to Hawaii, because Hawaii is now the only state you can travel to from Massachusetts without having to quarantine on your way back. The entire rest of the United States and the rest of the world is a red zone, according to Massachusetts, but you can go to Hawaii.
Alan, have you seen this DOD study that is referenced in the Washington Post article?
I have not, but that also looks interesting.
So I learned of this. I get the monthly United Airlines magazine that they used to give you on the plane, and they decided to send it to people because they're just piling up in warehouses, right?
Right. And
they said, we did this study with the Department of Defense where they took a plane and they put filtration units and they measured. Anyway, it's described there, and they conclude, now I don't know, I haven't seen the data, it's not been published, they conclude it's a really safe place to be because the air circulation is top down and there's HEPA filtration. And if you wear a face mask, they say it's one of the safest places you can be. However, of course, the airport is not so safe, so you have to get through that to get on a plane. So they say even if the plane is full and everyone's wearing a mask, there's low risk because they have measured. And there's a picture of a guy in this article with a pipetting next to these dummies sitting in the plane. I don't know what they actually did, but United Airlines uses this to say, hey, come fly with us. It's safe.
Fly with it.
Yeah. So I know that there have been a few papers that have come out about particular sort of cases of transmission on flights. There were a couple of emerging infectious disease papers and one in JAMA. But none of them actually looked at comparisons of how masks or how any other kind of mitigation strategies would impact that. So
we did cover those earlier on TWiV, I believe, right?
Right.
We did talk about those. Yeah, they were. Go ahead, Rich.
I got a hunch the most dangerous thing about flying is the airports.
Yes.
Probably right. Yeah. Now, a lot of people are sending letters and asking, is it okay to fly? And I'm not going anywhere, folks. Because
I would say it's not okay to fly because you shouldn't be traveling. So if people are wondering, gee, is it okay to fly to such and such to see my family for Christmas? No. And the flight's not the problem. The problem is that you're going to go to another household full of other people and expose everybody and sit around a table or what have you. Don't do that. No,
that's a good point.
Sorry.
Very good point.
Merry Christmas, but do it on your own.
Dixon, can you take the next email, please?
I can, but I'd rather have Rich do it because he sings much better than I do.
Oh, I'm not going to sing this because it's the.
You have to.
No.
This is a perfect
we got to show the YouTube thing.
Yes.
You want to show it?
Oh, okay.
Because they do such a good job.
Okay, fine. So I'll just read the email then.
Yeah.
Dear TWiV stars, it's 28 degrees Fahrenheit, minus 2 degrees Celsius, snowing huge snowflakes here in Santa Fe, New Mexico. Joe and I have loved all of the poems and limericks that you have read. So creative and wonderful. We thought you needed a song, so we created the COVID Carol, to brighten your holidays. We were recording music for holiday concerts in our Santa Fe theater, the Lensic. And since we had the sound and video crew set up, we took the opportunity to record this song written for all of our favorite TWiV friends. Happy holidays to all of you and to all of your listeners. There is hope at this season with the vaccine already being deployed and with the accurate scientific information that you give us in your weekly podcasts, the bright light that has kept us both sane and informed throughout this pandemic. It is with much gratitude that we dedicate this song to all of you. Hit it, Rich.
No, I think Rich is right. The video, we should just link to the video. This is
okay. Okay. As soon as I saw it, though,
if we're not actually going to show this, everybody. No, actually, I tried singing it. I can't do it justice. Okay.
I could hear you singing it, though, when I first read it.
Some of it I could do justice, but no, it really, the video's got the whole thing. And you guys, I assumed you watched this?
Yeah, of course.
Yeah.
Did you notice?
Yes.
That sitting on the piano,
of course,
they got your 2 volumes of Principles of Virology?
Absolutely.
I thought he ought to have one open on the music stand playing music.
Yeah, that would be funny. So I asked them for permission to post this on the Microbe TV YouTube channel. So I'm going to do that this weekend. So it'll get a lot of views because it's unlisted at the moment. But I sent this to the textbook authors because, I said, hey, this is pretty cool. There's your textbook on the piano there. And they really liked it. And I have to read Theodora, one of the co-authors of the textbook. She said they should have included these lyrics. And let me read them to you. She said, "Oh, my God, this is brilliant. They even got the virus versus the disease right, which most journalists don't. And the book on the piano, nice touch." She said, TWiV is awesome. I have a minor suggestion for the lyrics, though, in jest. After the names of the other hosts, she said you should add, "But do you recall the most cranky host of all?" Vincent, the Twitter prof, VRR, had a very sharp retort, and if you ever heard it, it was known to make you snort. All of the other TWiV hosts used to ask him to calm down. They wouldn't let poor Vincent justify his righteous frown.
Wow.
Isn't that
Very
great?
good.
That is great.
It's the same melody that she sung it to, right?
Yeah, it's the song is to Rudolph the Red-Nosed Reindeer. It's excellent.
It's a great song because she and Joe follow some of our memes on TWiV, right?
Yeah,
which
so
is
it's
great.
got some of the arcs in it. Yep.
Some of the arcs. Really great. This is great. Thank you, Gina and Joe. She wants to pronounce
I was glad to rhyme with Vixen because that's just warms the cockles of my heart.
Very good. Thank you so much. That was you, Dixon. You read that? Okay. So, Rich, now you can take the next one, please.
Danica writes, "Hello to all TWiV Avengers. Yes, Avengers is how I view you all. Avengers assembling weekly to battle misinformation and protect the world with truth and science." I do have a question for you all, and I'm not certain which TWiV superhero is best fit to answer. That doesn't prevent us from all jumping in simultaneously.
It's all true.
My question is about herd immunity. From what I understand, we do not have any evidence that the vaccines protect COVID-19 from spreading to other people. Correct. We only have data on efficacy for the recipient. And efficacy, this is my footnote, is measured in terms of whether or not you get disease and sometimes on how severe the disease is. Certainly, absence of evidence is not evidence of absence, however. Very good. If we find this to be true, that the vaccines only prevent the vaccine recipient from illness, but viral shedding still exists at a level that can cause infection in others, it seems that vaccines might not be the herd immunity answer that we have all, including Dr. Fauci, been touting. Put another way, mass vaccination would not prevent an illness from spreading or getting established in a society, just prevent massive rates of illness, which isn't really herd immunity. So my questions, is touting herd immunity premature? Or can we reasonably assume that mass vaccination, even with viral shedding, can achieve herd immunity? Or can we reasonably assume that viral shedding does not happen based on what we know with other vaccines? Am I misunderstanding how herd immunity works? No. I welcome all of your thoughts on this. I think the question does relate to how we communicate about the vaccine. The, quote, vaccines are not just about protecting yourself, it's also about your community, end quote, really seems to hold water if vaccination prevents infectious shedding. If this question has already been discussed, please let me know which episode and I will make sure to listen. I'm a regular listener, but also recognize that I might miss some important discussions on these topics. Thank you, Danica from Bellingham, Washington, and she's an epidemiology grad student who just finished her finals and is stoked to get caught up on all of her podcasts. So let me have a crack at this, and I'm sure you will all jump in. You're correct. There is no evidence that the vaccine prevents transmission of virus from one person to another, which it could do. It could be that the vaccination would make it so that that transmission would not cause disease because you'd have some level of immunity. So the bulk of infections would be asymptomatic. To me, and this is just me talking, it depends to some extent on how you define herd immunity. If herd immunity really means that the transmission of the virus is stopped, then no, there's no evidence currently that the vaccine is going to stop transmission. And the virus could continue to be transmitted. But can we talk about, this is a question. Can we talk about immunity to disease?
Okay, so as herd immunity is, my understanding of the common definition is that it is it's a collective phenomenon that arises when a certain percentage of the population is immune from transmitting the pathogen. And therefore, the remaining percentage, whatever it is, depending on the pathogen, is protected by herd immunity, meaning the pathogen can't get to them because it can't find enough susceptible hosts. So if 99% of the people are vaccinated against, or whatever, are vaccinated against measles, then the remaining 1% who are not immune from measles will be protected because the measles virus gets nowhere. Right. It just can't find enough susceptible hosts. There's not a high enough concentration of susceptible hosts to spread it to the people who are susceptible. So that is the standard definition.
Does the measles vaccine actually prevent infection?
To an extent.
I asked Sally Permar, right, Brianne, and she said, yes. No, it's not sterilizing. Right.
No, but it does reduce transmission,
the load,
doesn't it?
Yes, it could reduce the load enough so you don't get effective transmission. That's correct.
Right. And that is in fact the rationale, and what we've observed with measles vaccine is when you have a high percentage of vaccination in a society, you don't see transmission even with occasional introductions. Right. Otherwise, we never would have gotten measles to near eradication.
No, it's all about transmission. If the vaccine doesn't prevent transmission, then herd immunity is not possible, right? But we know with many vaccines, it is possible, right? And so we hope that this one will convey herd immunity through impeding transmission, but we don't know. And yesterday, Daniel said that Moderna is actually planning a transmission study of their vaccine next year on college campuses. And that will be very interesting.
Yes.
Right.
And all of the companies who are doing these are testing for virus infection. But as I think there's a letter coming up later from somebody who's in one of the trials, it's not like everybody gets tested every day to see if they've contracted the virus. It's basically symptomatic surveillance because that has been the emphasis, is on preventing symptomatic disease, preventing COVID-19, the disease. But they are, when people develop symptoms, they're testing them for SARS-CoV-2. They're repeating the testing. They're checking, when you clear it. And so there are data suggesting, some things about transmission, but there aren't enough data to say one way or another it does or it doesn't prevent transmission. So we don't know.
Right. And so two other points to that. If you look at the Moderna data that was part of the FDA discussion yesterday, the supplemental table 1. So, when you're going through your 90 some odd pages of data, I'm sure you noticed supplemental table 1.
Oh, yes.
Yes. It did look at so basically between shot 1 of the vaccine and shot 2, people were tested for infection, and really they were testing so that if you got infected between those 2 shots, you got excluded. But they happened to have, as a result, they had data on, did the first shot of that regimen protect against infection? The numbers are quite small. And so I wouldn't, I'd take this with a Tonka truck full of salt, as someone said on Immune.
Tonka?
But yeah, that's what they called it. But it said that there was a difference of 67%. So there was 67% efficacy in protecting. And in the there's a New York Times piece by Apoorva Mandvilli that's called "Why Vaccinated People Still Need to Wear a Mask," and she asked Pfizer and Moderna if they were planning to use the blood serology samples that they had taken to do some anti-N antibody responses to look and see if participants had been infected. And they said, at least to her in that interview, that was planned.
Yeah, we have to see. We don't know. But let me just say that based on what we know about common cold coronaviruses, you have immunity. Every year you get infected anyway, and you transmit it. You don't get sick, but then again, they don't make you sick in general. So even your first infection as a kid, you don't get very sick. But you get reinfection, you get transmission of common cold, seasonal coronaviruses, even in the face of population immunity. So there can be,
and
the
we
key
know that
is, go ahead.
We know the SARS-CoV-2 can do the same thing. We don't know how often, but it does. We do have reliable reports of reinfections.
Yeah, but they're pretty. The numbers are low, so I don't know.
The numbers
if that's
are low
trend.
because in order to get a reliable report, you've got to sequence the whole darn thing both times, and that just hasn't been done very often. So we don't know how common
So I think
is.
the key to herd immunity is you can have shedding after vaccination as long as it's not enough to transmit. Transmission is really the key. You have to not just look for because if you do a shedding experiment after vaccination, I would say, what do the data mean? If you shed 10 to the 2 PFU per mil of nasal wash, is that enough to transmit or not? You have to look at transmission, and that's why the studies that are planned are important. I don't know
and
what
that's
to
why
predict.
Something like the measles vaccine, it's not sterilizing immunity, but it's good enough.
It's good enough, yeah.
Until maybe late in life, it wanes in some people, but if you get the percentage of the population, but then you look at something like the inactivated polio vaccine, where you can perfectly well act as a carrier after you've gotten IPV.
Absolutely. And there have been cases where that virus can spread in the intestines of people who have IPV, and they don't get polio. But then if it gets into a person who's not been vaccinated, they will get polio. Yeah. They're not protected because the virus is still transmitting.
And for flu, my understanding is that it varies year to year. There have been flu years where the vaccine offered pretty good immunity against transmission, and there have been years where it offered not so hot immunity against transmission. And then on illness, it's kind of scattered as well.
Yeah. We have this question multiple times in the emails, as you will see, but
this is a critical point, and it's okay to think that the data don't answer it because they don't yet.
Yes, Danica, one of the things I've been planning to do for a while is take a deep dive in doing some reading on some other vaccines on this exact point, and I just haven't had that happen quite yet.
All right, Andrew writes, I trained at, this is Andy. Hi, Vincent. I trained at New York Hospital in the late 1980s, so maybe we even passed each other at the PK. That's the Papaya King. So Andy sent in the picture last time of the Papaya King on 86th and 3rd Avenue. In case you didn't catch this, an EUA was granted today for the first over-the-counter, fully at-home diagnostic test for SARS-CoV-2. And he provides a link to the FDA announcement. Today, the FDA issued an emergency use authorization for the first OTC fully at-home diagnostic test for COVID-19. The Ellume COVID-19 home test is a rapid lateral flow antigen test. It runs a liquid sample along a surface with reactive molecules. It detects fragments of proteins of SARS-CoV-2 virus from a nasal swab from any individual 2 years of age or older. Would be interested in hearing the TWiV team and Michael Minna's take on this news. All the best, Andy, is from the FDA. Now, Daniel talked about this yesterday on the clinical update in quite some detail. And he said that they recommend you do a brain swab, what are those deep nasopharyngeal swabs all the way up. And he said, it's going to be interesting to see how that works out, because I'm not sure you could put it all the way up your own nose, right? I certainly couldn't, because I guess there's more antigen up there than there is at your anterior nares. I mean, it's a good start, right? This is about $30. You can buy. I don't think you need a script. And Daniel said that's good. You shouldn't have a physician standing between you and a diagnostic test. So we'll see if it works out. And I'd like to get one. I'll see if I can get one. But I'm not sticking the thing all the way up my nose.
Yeah. And $30 a pop is going to be, I mean, that's a lot for a lot of people. And that's certainly a lot for anybody to be doing on a frequent basis.
Yeah. Now, since Michael Minna's name is here, Michael Minna and some other people have an op-ed in the New York Times today. And I find it, what's the word I'm looking for? Not a good idea to have published this, okay? He's saying that, hey, there seems to be some efficacy after just one dose of these mRNA vaccines. Maybe we should just give everyone one dose and vaccinate more people. I think that's the wrong message to send.
No, terrible idea.
There's a little bit of efficacy, but it ain't good enough. You need 2 doses. And why are we suggesting that it would be a good idea to have only one? I don't get that, Michael.
No, and the EUA came through for a 2-dose regimen, and it's going to come through for a 2-dose regimen from Moderna, and that's what the trials were done on, and that's how the vaccine works. And you don't muck around with that at this late stage of the game just to accelerate your vaccination schedule by a few weeks, most likely. I mean, the companies are gearing up to produce millions and millions of doses of these, and it'll proceed at the pace it proceeds at.
Yeah. I remember being very strongly impressed by the effect of a boost on at least antibody production in the phase 1 and phase 2 trials.
Yeah.
And my reaction to that every time we saw it was, you need a boost.
Yeah.
Right.
Brianne, can you take the next one?
Sure. Aaron writes, good morning. There's an FDR quote, when you reach the end of your rope, tie a knot and hang on. Your thoughtful, patient, and wise voices have been my knot. Rich Condit's gentle bloom and Brianne Barker's clear diplomacy, Vincent's salty crackle that cares, and Daniel's direct dispatches from the front lines. All of you combine to bring me a sense of peace and trust that we are in the good, capable, and dedicated hands of experience, even when the chaos of Washington seems otherwise. Thank you. You have also been kind enough to answer a question from me a time or two, and now I hope you might answer one from my 5th grader. He was thrilled to hear that we will have a vaccine, dampened slightly by realizing that manufacture and distribution will take time, but positively crestfallen that kids aren't on the list. He wants to understand when and how the phases of development will happen for kids. He is ready to have sports, sleepovers, socializing, travel, and all the hugs back, aren't we all? Thank you as always for your guidance, a mom in Texas. So the trials were done as some people have discussed for Pfizer for people who were ages 16 and up. And then after that, there are additional studies where the ages are de-escalated or something like that. I can't remember what word they use, where basically people of younger and younger groups are included in the trials in order to ensure that the vaccine is safe in them. And those are underway right now. Though I do not know the specific ages or times.
Both Pfizer and Moderna are doing trials in kids. I think it's ages 12 to 17. And I think the original Pfizer trial, at least the data were only down to age 18, but it got approved in the US under the EUA for down to 16. There was a little bit of a discussion at the FDA about that. But 12 seems to be the next kind of target point. And then I guess from there, they would go to younger groups after that. The idea is to sort of edge your way down to see if there's some pediatric issue, because you really, really don't want to mess that up. So a fifth grader is going to be turning 12 soon, I assume, right? Going to be...
I think you turn 11 in grade.
Sixth grade.
Sixth grade. Okay. So if a fifth grader now, by the time, I'm not expecting anybody to have normal school in the spring semester. By the time fall semester is starting, that'd be sixth grade. And if they're coming up on 12, then they'd be potentially getting vaccinated at that point, maybe, if these trials go well that are getting started now in the 12 and up set. So yeah, I would say, look forward to sixth grade, maybe. I was thinking
about this issue the other day, and here again, I'm talking without any information. I'm making this up. I'm posing this as I'm asking you guys for your input. It seems to me that having proven efficacy in a large population in adults and safety in the same populations, that the major concern in kids is going to be safety.
Yes.
And you're not going to need 40,000 kids to prove efficacy. And in fact, most of the initial safety trials in phase 1 and phase 2 are done with smaller numbers of people. So I'm kind of assuming that as we reach down to get younger and younger populations, that this can happen with fewer individuals and on a quicker time scale. Does that make sense?
Yes. On that same episode of Immune that Vincent mentioned earlier, Sally Parmar talked about this a little bit and had some ideas of how quickly it might happen. I don't remember all of it right now, but people can go back and look at what she was saying on that front.
So I just found an article in The Guardian. When can kids in the U.S. get the vaccine? And I don't know where they get their information from, but they say it doesn't appear that kids will be able to get it in time for the fall semester next year, based on the timeline. I don't know why it's so long in their estimation. They don't say why. Maybe they're basing it on the current length of time it took for the, these vaccines. But as Rich said, I think it can be done faster. So that surprises me.
We'll see.
Yeah.
Good question. Really good question.
So the next one is from Justin, who sends a link to a video he made. He apparently got his small rural EMS agency got the vaccine. He made a video of him bringing it around and getting it or giving it. So we'll post a link to that. And Alan, can you take Lori's, please?
Sure. Lori writes, I really have enjoyed TWiV and the great information you're discussing. I'm hoping to take advantage of the holiday season to mobilize an on-site COVID rapid testing for my son's high school. Now that there will be over-the-counter testing available, I believe we could have a high school organized to get the kids to do the test, interpret them, document, record, and report results. I have tried to involve the bureaucracies, government, education, health. I'm not sure they will help, but let's assume I can get this going. Can you tell me what test you recommend for this? And is there anyone that knows how to order these in bulk? Studies I have read recently suggest all teachers and students tested every 2 weeks can adequately keep our environment safe and kids in school. Thanks for any input. Well, there's the over-the-counter test we just talked about at $30 a pop. I don't know how big your school is, but I'm going to guess that you doing every 2 weeks at $30 a pop on a high school of 2,000 students is going to be probably more than the PTA has raised this year. I'm just guessing. It's going to be an expensive process. But depending on the resources available, that's maybe a possibility. It has the advantage of being approved for over-the-counter use by non-medically trained people, which could be could make a big difference if you're hoping that students and staff can do it themselves.
So the other ones, Alan, the quick lamp assays,
right.
They require trained personnel to do them.
Yes.
Yeah. So that's the only one, and I think though others will come out, But $30 is expensive. No lick of sticks yet, unfortunately.
Nope. the LAMP test, could that be part of the nurses' training in the school itself?
Could be.
Do schools still have nurses?
They do.
Yes, they do.
I mean, that's possible, but it would be. It might be a stretch, and with the LAMP assays that I'm familiar with, you need a device to process and interpret the results, and so that would be an investment the school would have to make. And again, that's not necessarily something that they're going to pony up for.
There's a LAMP test that's very small now. It's about $30.
Right. Yeah.
But that also requires a prescription or trained personnel.
Right.
But Lori, listen to the previous TWiV 695 by Daniel. He talks a little bit about that, the different tests that are available. Dixon, can you take the next one?
Sure. Anne writes, Dear TWiVers, like another of your listeners, I'm in a Regeneron clinical trial where 4 subcutaneous shots of the antibody cocktail are given sub-Q monthly for 6 months to healthy people. I've gotten 3 sets of shots so far. I'm 70 years old, and at that site I do go to, the injections are typically 2 in the abdomen and 2 in the thigh. 75% get the antibodies and 25% get the placebo. I have no idea of which I am getting. With the vaccines coming out, I might contemplate dropping out at some point to get a vaccine, especially as the trial continues 6 months past the administration of all shots to continue blood draws and such. But if I do, I have a dilemma that I hope TWiVers could help me with. I have to assume I'm getting the real thing, and if I am, from what I understand, I'll want to wait for a period, at least the Regeneron antibodies interfere with the effectiveness of a vaccine. But for how long should I wait? I assume it depends in part on the half-life of the antibodies, but I've been unable to figure out how long that is likely to be. And once I know, how long do I wait? 1 half-life? 2 half-lives? Any thoughts or help from the TWiVers will be much appreciated. Regards, Anne from, Rich, Austin, Texas.
So let me just jump in and say, Daniel addressed this on TWiV 695. He said exactly how long you have to wait after you get the monoclonals to get vaccinated, because there is some concern that there'll be interference. So I don't remember what he said. 695, okay? Anne, it's only 40 minutes long. You can find it.
Good question.
It's a good question.
Excellent question. Yeah.
Yes.
Rich, can you take the next one?
Scott writes, greetings, TWiV team. While I understand the definition of infection and disease with respect to SARS-CoV-2 virus and COVID-19, I do not really understand how someone who is vaccinated can shed virus and infect other people. It seems to me that if you're vaccinated, your body should be able to stop the replication of the virus and prevent it from spreading to other people, but clearly it is not so. So I guess I would like to better understand how one can be infected and shed virus, but not have it cascade into disease, sickness. Could you potentially elaborate on this? Regards, Scott. He's from Menominee Falls, Wisconsin, where it's cloudy and below 0 degrees these days. So nothing in this business is black and white. You can tamp things way down to the point where you don't get disease, but that doesn't mean that there's no replication at all. That's the way I think of it. And you say the vaccinated should stop the replication, prevent it from spreading, but clearly this is not so. Actually, we don't know the answer to that question yet. Okay. We don't know whether or not it's going to be what we call sterilizing immunity, but I think you can have some sufficient immunity to prevent disease without necessarily preventing the virus from replicating. And I would point out that if you stick around and listen to TWiV post-COVID, you will find out that you are crawling with viruses that replicate and don't cause disease.
Crawling, I like that.
Yeah.
Alan, did you take one in this last round? I don't remember.
I did. I just took.
Okay, you did.
I did take. Yeah.
Dixon, you did also.
No, you skipped me. That's okay.
Dixon did not.
Oh, you just did the.
The one before that, the cycle before that. That's
all right,
okay.
We'll get you. What were you going to say, Alan? Though before
no worries.
Right. So one possibility with a respiratory virus, for example, could be that you have antibodies circulating in your bloodstream, but you don't necessarily have antibodies in the mucosal membranes of your nose and your lungs,
Sure.
Which would be in a different class of antibodies. And so you may be able to replicate some virus in those places, and then it gets detected in the bloodstream, and then your immune system kicks in and takes it out. But it may be there long enough to transmit to somebody else.
Give us an example, Alan.
That was the one I cited earlier with the polio vaccine.
Got it. What a good student. What a good student.
If you get the virus or you get the oral vaccine, you get mucosal immunity. You get, in that case, it's a gut virus. So you get immunity in your gut with immunoglobulin A, and you will not be able to carry the virus after that. You won't be able to transmit it. But if you get the injected, inactivated vaccine, you get the IgG antibodies in your bloodstream. You don't get the IgA in the gut. So then when you get polio virus exposure, it replicates in your gut. It goes out in your feces. It goes and infects the rest of your third grade class. But it can't get into your bloodstream and then get the viremia that causes the paralysis. Instead, your buddy, who went down the slide after you gets it, and they get polio.
Are you implying that kids don't have good hand hygiene, Alan?
Oh, no, I wouldn't suggest that.
All right, Dixon, can you take, Maureen? Yes.
Yes, I can. Maureen writes, love your show. In med school, immunology was my favorite basic science, but my life work was in family medicine. I'm now retired, but fascinated with COVID. I heard last week that herd immunity is not possible with messenger RNA vaccines because they only prevent disease, not infection or transmission. Gee, that sounds redundant. So we need 100% immunization for all to be protected. Tonight, I heard Dr. David Kessler on the news mention possible herd immunity at 75% vaccinated. So which is it? Does the vaccine prevent infection and transmission or just disease? Maybe I misunderstood. Rich, take it. Please explain on your show. You may have today, but I haven't listened yet. Thanks so much for what you do. Maureen from Juneau, Alaska.
Wow.
Nice.
Far away.
I've been to Juneau.
How cold it was.
Yeah, you have?
Yeah, a long time ago.
I've never been to Alaska.
I haven't either. I'd love to go there. That's on my bucket list.
Rich, you want to grab that?
Hey, we don't know yet. Okay.
Thank you.
And
that's exactly what I thought you'd say.
Yeah, we know it prevents disease. We don't know if it's going to prevent transmission, and the answer to that is probably going to influence really what percentage of the population is really going to need to be vaccinated in order to achieve what we call herd immunity, and refer to the earlier parts of the show, which Maureen, you've already listened to.
Yeah.
So the statement that herd immunity is not possible with mRNA vaccines is not right because we don't know yet. But Kessler said it could be possible at 75%. Well, if it blocks transmission, then yes, that's the number that we figure from the R0 that would give it. So he says possible, which is correct.
Right. But that's predicated on what we would call sterilizing immunity, 100% blockage of transmission with the vaccine. Then you'd need to vaccinate 75% of the population, according to the calculations, in order to get this protective herd immunity effect for the remaining 25%. What we were saying on the last episode is if you have 0% immunity against spread, then you would need to get 100% of the population. I don't think either of those extremes is going to be the case. But the point that was being made on the previous episode was those extremes exist, and we're probably somewhere on that spectrum, and we don't know where. And so we need to prepare for the possibility that this vaccine may not protect against spread as well as we'd like it to, or it may.
And that's why you need to keep listening to TWiV, because we'll discuss all of
this.
I
was about to say that, actually.
I think you should keep listening. I'm getting very sad at the specter of all of you leaving.
Yeah.
Alan, your definition of herd immunity earlier in the show was really good. It's the level of immunization required to prevent spread to people who have not been immunized.
Right.
Transmission. Transmission is key.
Transmission. It's a population-based concept. It's not an individual thing.
Here.
All right, one more. Sarah, thank you for all your work. I'm one of the newbies who started listening in January 2020. Cannot express enough how incredible it has been to be so informed during this pandemic. Just think, so we have 100,000 listeners, maybe more. It's like, what fraction of the U.S. population is that? It's pathetic that more people don't learn from us.
But the influence of the show goes beyond them, because we frequently hear from people who are the source for their families when they have questions about stuff, and we're the source for them. And so,
I guess so.
I think the reach of this show is quite large.
I can't do anything about it, but I can complain. Isn't there a saying about that? I even love it when I hear terms and processes that I don't understand. There's something very reassuring about it. Hey, isn't that someone said they love hearing people talk about things they don't understand, but it just sounds good? No, I said that. That was the video, my pick a few months ago, the guy explaining some music theory. I didn't understand most of it, but I loved hearing him talk about it. I get that.
Sarah, that's why I show up every week, Sarah.
Dixon, you are a scientist. In fact, you work on infectious diseases, parasitology.
Yeah, I know that.
So
but there's a lot that you guys say that I don't understand. I must say that.
Dixon, there's a lot you say that I don't get.
Yeah, but that's different.
Like you say, although I did subsequently learn, you used to say ecotone all the time, and I didn't know what you were talking about.
I've eliminated that from my vocabulary.
Well, I learned what it means.
Yeah,
I learned what it means. All right, so Sarah wants to know, if you had the choice between Pfizer and Moderna vaccine, which would you choose and why? So whichever first was offered to me, Sarah, doesn't matter.
That's right.
Rich Condit, which would you take?
No preference. I'll take whatever I can get. Same thing. First thing.
Brianne.
Yes, please.
Great. Alan Dove.
Me too.
Dixon.
I got one on each arm. Come on, right here.
Yeah, either one. They're both the same, more or less. One has stability differences, but it doesn't matter.
And it's not, I mean, if something comes out later where we find out one is superior over the other, then I might develop a preference. But from the data we've got from these large phase 3 trials, it's do you want to buy a Honda or a Toyota? You're not going to go far wrong.
It's the identical sequence for a messenger, right?
Right. Messenger RNA is the same. The secret sauce, the lipid envelope, is proprietary to each. So there's a difference between the two. And the storage temperature may have something to do with that. The Moderna vaccine is a little easier to store, at least from what we know so far.
Certainly the efficacies were both, high,
efficacies were
mid-90s. They're fine.
Yeah.
Now, if you ask me about the adeno...
right now,
what?
I'm sorry to interrupt. I was going to say the thing that's killing me right now about the Pfizer vaccine is that they've got a warehouse full of it, and the government hasn't told them where to send it.
Is that right?
That's right.
Pfizer. That's how you say it, Pfizer. Dixon, if you had a choice between a Tesla and a Tesla, which would you buy?
I'd take the second one.
Dixon, by the way, what's your pick of the week? Let's do some picks.
Okay, I'm glad you asked, because you know me, I'm a visual kind of person. So I looked around for the annual contest winners for a variety of websites, and I found a stunning set of bird pictures from the Audubon Society for the award winners for 2020. Some of these, I mean, I cannot believe how difficult it is to get those pictures. You can sit for hours, days, months, and never see any of this. Wow. And one of them, which is the hummingbird that is actually drinking a falling drop of water, is for me, that was the winner, bar none. I don't care what other pictures you showed me. I think it's just so endearing and warm and nice to know that wildlife is still out there and behaving normally.
I feel bad for this roadrunner. He's got his hand there. He's trying to get out of the beak. Poor guy.
You mean the lizard?
The frog. The lizard, yeah.
No, Roadrunner, it says, oh, that's the bird. That's the bird.
Roadrunner is the bird.
Yes. What is the thing? A lizard of some kind, right?
Yeah, a lizard of some kind.
A desert animal of some sort.
It's funny. I follow on Instagram a lot of people who take bird pictures, but I do not, and I like all of them. I hit like, but the ones where a bird is eating something, I never like them because I can't like it. I know it's nature.
Even a seed?
What's that?
A seed? Come on,
A seed, if it's eating corn or something?
No, if it's eating another
animal.
Another animal.
Life form, another life form.
Like a lot of people take pictures of hawks ripping apart squirrels and mice, and
It's true.
I know you have to do it because you can't go to the supermarket, but it's just I can't like it. I'm sorry. It's just.
I did see a picture once of a praying mantis that had camped out on a hummingbird feeder and caught a hummingbird.
Yes.
Really?
Wow. I saw that.
These are gorgeous. These are so sharp, Dixon.
I'm glad you like them.
Really cool.
Amazing.
Glad you like them.
I love the one of the bird drinking too. That's just beautiful.
Yes.
Just birds are amazing. I really like birds. And the one with going in the water with the splash.
I like the diving one at the top.
Yeah.
So cool.
Yeah, the cormorant.
It's all great. It's all great stuff.
Brienne, what do you have for us?
Oh, so I have latest piece by Ed Yong, who is my favorite science writer who's not Ellen Dove. And
it's okay if he's your favorite, bar none. That would be all right too.
It has actually two different titles depending on where you look. So I first saw it listed as COVID-19 Changed Science Forever. When I was putting it here earlier, it was called How Science Beat the Virus and What It Lost in the Process.
I like that one better.
Yeah.
Yes. This is a long form piece all about kind of changes in science, both in terms of what we are doing differently. It talks about preprints and the rise of preprints and what that is doing in terms of publishing. It's talking about how many people were pivoting their research, but other types of research that was shut down. It talked about, the new ways that we're able to make vaccines so quickly and how that may change vaccines in the future. And it's a really good overview of a lot of the things that we've been talking about throughout this whole pandemic, and just general changes that we see in science. And I found it to be really fascinating. And I think that people will really like this as a nice overview. Plus, Ed Yong's writing is just always lovely.
Yeah.
However, I think he's right, but he doesn't know the half of it because he's not in there. No, the people he talked to, but there are a lot of other stories. Absolutely.
Oh, of course. And there's also, stuff about equity in terms of who's having, trouble moving forward in science, who's not. He has a nice quote about from Dr. Fauci about the virus was fully sequenced in January 2020, and now in the fall we're finishing a phase 3 trial. Holy mackerel!
Yeah,
it's pretty cool. Alan, what do you have for us?
I have a book that I read just recently. It's called Entangled Life by Merlin Sheldrake, who has a surpassingly cool name, and this is a very fun book. It's about fungi, and the some of it I knew as a microbiologist, a lot of it I didn't. The integration of fungi in ecosystems and in our lives and in civilization, and it's just it follows all of these threads like intersecting hyphae woven through everything that you can imagine. And there's a phenomenon. There's one that he comes back to a few times that's sometimes referred to as the wood wide web, which is if you go into an established forest, the trees are all connected to each other through fungal hyphae under the ground, which turn out to be a communication system and a nutrient exchange system and almost a marketplace that the trees use to exchange nutrients over various seasons. And it has a profound effect on the entire ecosystem. Nobody even knew about this until like 10 years ago. It was this, and it's amazing the extent to which this is there and has always been there, and what is it doing? And as we find out more about what it's doing, it just gets more and more bizarre. And if you haven't spent any time reading about fungi, be prepared to have your mind blown. And if you have, this will also be cool.
So
really
Alan,
fun book.
The magazine section of the New York Times last Sunday featured on the cover a picture of a forest, and then the whole article was about a woman by the name of Suzanne Scott.
Coincidentally, I just finished reading this book, and then there was a Times Magazine article that was about what I'd just been reading in this book. Oh my God, the fungi are coming to get me.
You should see her TED Talk. I watched it.
Okay.
It is phenomenal. It is absolutely phenomenal.
How about if we listen to it?
You could do that too.
You could do that too.
Did you know that Elio Schechter wrote a book about mushrooms and the company of mushrooms?
Oh.
Yes, he's an avid mushroom collector.
How old is he?
Just 90-something, 92 now,
usually,
I think.
Mushroom collectors don't live long lives, usually.
No, good ones live long lives. It's fine.
Well, he said it's harder for him to get out now and... well, because now he's not going out at all, but he said it's harder for him to get around and collect. And he said, if you don't have rain, you don't get many. And they haven't had a lot of rain in San Diego as well.
It's true. It's true.
Rich, what do you have?
So I am a regular follower of PolitiFact, mostly because there's always something in our daily paper. PolitiFact is a website basically that started out as a nonprofit project of the Poynter Institute in actually started out with the Tampa Bay Times. It's now the Poynter Institute in St. Petersburg, and they evaluate statements and publish statements from politicians, elected officials, candidates, staff, lobbyists, and evaluate them for accuracy. They have a truth-o-meter rating that goes from true at one end to pants on fire at the other end. And this year, as in many previous years, this being the end of the year, they published the lie of the year, which is my pick. And the lie of the year is the coronavirus downplay and denial. And it's a nice article that goes through with a bunch of links to back up their assertions that basically says that the downplay and denial of coronavirus has been a disaster. And they point the finger at our outgoing President Trump as being a major perpetrator of this.
But not the sole perpetrator.
Right. But there are a bunch of other collaborators, including news organizations such as Fox News and other sites. And you can read all about it here. And this is, as Vincent said earlier, not politics. We're talking about what's factual and what's not. And the bottom line here is that this lie has killed people. Tens, hundreds of thousands. Okay? So this is not a trivial thing, and you need to pay close attention to it. And before I finish my rant, I have to say that I woke up this morning thinking about this and thinking about how truth and lies have been, with humanity forever. And I was trying to think of an example, and I came up with what turns out, I knew it was New Testament, turns out to be John chapter 18:38 in the New Testament. This is when Jesus is on trial and is facing the governor of Judea, Pontius Pilate. And Jesus basically tells him that his purpose is to bear witness to the truth. And Pontius Pilate is famously said to have said, "What is truth?" And I just point that out as a philosophical evidence and a philosophical question and evidence that this has been with us for as long as people have been around. And that's what we try and do, is bear witness to the truth.
Cool. It's really horrible. It really, the lie is horrible.
It's horrible. It's horrible.
All right. My pick is a article in today's New York Times, how New York City vaccinated 6 million people in less than a month. And this is, 1947. A guy walked into New York. He got sick. Turned out he had smallpox. So someone in New York City, the public health, decided that everyone should be immunized. And they did it. And they have great pictures of people lining up to take I should say vaccinated. Amy says, Vincent, you should say vaccinated, not immunized, because not everyone makes an immune response. Okay. Vaccinated. And it's a great story. A lot of cool pictures in this. The glass capillaries containing the smallpox vaccine, so forth. But it really is funny because in 2009, I wrote the same story, basically. Smallpox in New York City, 1947. I included the actual New York Times article, the single column article that was it says smallpox in city inoculation urged. And I wrote, millions of New Yorkers were immunized within a few weeks. While we are in the middle of a massive influenza immunization campaign, it's useful to review the accomplishment as a model for public health planning and mobilization. Those were my words. December of 2009, we're immunizing against pandemic influenza. So I thought it was cool how the city said, we're going to do this, and people said, okay. And now, of course, the Times has rediscovered this many years later. So I think it's a great story. One sick person walks in. Mm-hmm. Wow. And different today.
Yep.
Different response today. It's very
Vincent,
cool.
Well, also, we had the vaccine then.
Oh, yeah,
so
of course. But
it was a much quicker process.
Even then, people said, okay, they did it, right?
Yeah.
Right.
And I think as the vaccines roll out, a lot of the people who said in telephone surveys that they weren't going to get the vaccine are probably going to get the vaccine.
I hope so. Yes, Dixon.
Remember, on the corner of 168th Street and Broadway is the Public Health Department's station for diagnosing diseases, et cetera. And I grew up in that building. I was there from 1962 to God knows when. And I remember the sign outside that says, no shots, no school. And everybody took their kids in for vaccinations as a result, because there was a lot of immigration during that time from various places throughout the Caribbean. And they didn't have shots, or they didn't know. You can't go to school unless you've had their vaccinations, and everybody lined up. And you should see the lines going around the block. It was like some rock show was going to open.
Yeah, this article in the Times has pictures of lines and lines of people snaking all over the place,
Exactly.
Really cool.
Exactly. Thank God it's not for the COVID vaccine, right? That's the way to catch it. Stand online for the vaccine.
Well, you have to be masked and 6 feet apart.
6 feet apart, yes.
Yeah, good luck on that. Everybody's definition of 6 feet seems to vary quite a bit, though. Have you noticed? You go to a supermarket, they now have the actual places where you must stand.
Dots on the floor,
People here too, in the elevators. Dixon, in the elevators, you haven't seen them, but they have dots on the floor where you're supposed to stand. Okay.
In our apartment, you're not allowed to get into an elevator if someone else is already in it.
Oh, yeah? Yeah,
we've got two elevators and a freight elevator. You can't do it.
We have 4 per elevator, but that's too many because you're only 2 feet apart at 4. That's not right. I didn't make up those rules. All right, that is TWiV 696, microbe.tv/TWiV for the show notes, questions and comments, TWiV@microbe.tv. And if you like what we do, consider supporting us, as many have done so far. Microbe.tv/contribute. Dixon Despommier is at Racaniello.org and thelivingriver.com. Thank you, Dixon.
It was marvelous, absolutely marvelous.
Alan Dove is at Turbid Plaque. No, he's at alandove.com, and he's Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker's at Drew University, BioProf Barker on the Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently living in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.